So the second part of the lab 4 deals with the protein assay. Okay, so if you have a sample made with DNA or RNA, yeah, it's straightforward because you can do the spectrophotometry assay using a UV light of 260 to quantify the concentration using the reference points, and then the ratio of 260 over 280 will give you idea about their purity. That's straightforward. That's possible because DNA, RNA, there isn't much structure variation among their sequences because DNA, RNA, they are made up with nucleotides. DNA with GATC, only 4 nucleotides. RNA with GAUC, only 4 nucleotides. So there isn't much variation in their absorption capability, right? But if you think about the protein molecules, okay, so protein molecules are much more complex. They are much larger because they are made up with amino acids that are larger than nucleotides. And then their spectrophotometry absorption profile are more complicated. Okay, it depends on what amino acid you have. Their absorption capability are widely different, especially the amino acid with aromatic ring structure. Yeah, they can absorb the light, UV light pretty well, but other amino acid molecules, they don't absorb UV light very much. So depends on the sequence of your amino acid. Proteins, they don't give you a very straightforward UV absorption profile. So that's why you cannot use UV absorption in your concentration assay. Okay, so instead you use a color or dye, okay, that can bind with DNA, bind with protein molecule, and then use that color dye molecule in your spectrophotometry, because there should be a correlation between amount of color dye that is stuck on your protein, and then based on that color chromogenic substance, you can detect their concentration using a visible light. That's how you do this. Okay, so among several different possible color-based protein assays, you're going to do a lab exercise on an assay called Bradford protein assay. Bradford is the name of the inventor who made this dye, of course. So Bradford assay. So basically Bradford assay has Comasi blue, brilliant blue, that's the dye, blue dye, that'll stick to your protein very well. Okay, once it is stuck on your protein, then if you shine visible light, yes, the absorbance pattern profile will depend on the concentration. It correlates with the concentration of the protein you have in your sample. Okay, that's how it's going to work. Okay, so especially between this range, 5 and 50 microgram per mil concentration, you see the correlation between the concentration and absorbance pattern. Okay, so you're going to use that to quantify it. So especially the sample, unknown sample you are given with, is made up with a protein called bovine serum albumin. So bovine means what? A cow. Serum albumin is what? Blood protein. Okay, so it's a cow blood protein. It's purified. So Zach, our lab assistant, prepared some unknown sample for you, and then you are also given with stock solution of BSA to create a standard. Okay, so you're going to have to deal with the standard curve first, and then you're going to estimate the concentration of unknown sample. So the first part of the assay, okay, is based on you to construct the standard curve, okay, on your BSA's sample. Okay, so you are given with a stock solution of a BSA, bovine serum albumin protein, that has about this concentration, 10 microgram per microliter. That's a highly concentrated stock. Okay, using this stock solution, you're going to create a series of diluents. Okay, like you did on your endotoxin assay. You're creating multiple test tube with different amount of BSA protein. First one would not have any BSA, it's just water only. And the second tube will have about small amount of BSA, maybe 2 microgram per maybe milliliter or something. Okay, and the next one will have 5 microgram per milliliter. And the third one will have 10. Fourth one will have 20, and the last one will have 40 microgram per mil. Okay, that's the concentration. So to create those serial diluents, then how much of your stock BSA stock solution do you need to add to each all these tubes. The first one, of course, you will not add any BSA, so it's going to be 0 because it's just water only. How about this tube, which will have 2 microgram of BSA only? So your stock has 10 microgram per microliter concentration. So 1 microliter of that will give you what? 10 micrograms. So it's about 0.2 microliter will give you 2 microgram, right? And this one will be 0.5 microliter. That will give you about 5 microgram, and this will be 1, and that will be 2. That will be 4 microliter of the stock BSA. Then you will get that much of BSA added to all these tubes. Okay, and then how much of water do you need to add to make 800 microliter? So here it will be 800 microliter, right? And here will be what? 899 point maybe 8, right? And that, I'm sorry, not 800, I'm sorry, that's 700. And that will be what? About 799.5, of course. And that will be 799. And next one will be 798. Oops, I'm sorry. And then that's 798. And the last one will be 796, right? So these microliter of water, if they are mixed up with this much of stock BSA solution, you will get 800 microliter total if you combine these, right? To those tubes, you will add 200 microliter of BSA assay solution. Okay, so what you need to do is you're going to probably add water of these volumes to all these empty tubes first, and then you're going to add your stock solution of BSA to all these tubes. Okay, once these two are mixed, water and BSA is mixed, and then you're going to add your assay solution. That should be added at the last step. Okay, so by combining BSA assay solution with 800 microliter your mixture of BSA and water, you will get 1000 microliter volume there, right? And then these mixtures will be transferred to a cuvette. Okay, cuvette is a special sample container that doesn't absorb light by itself. Okay, and then you're going to transfer these samples to the cuvette, and you're going to do the spectrophotometry reading inside of using the sample in the cuvette, right? So you're going to write down the observance here. Okay, and then the first the sample which did not have any BSA will be used as your blank cuvette. So blank cuvette is needed when you do any spectrophotometry reading, because that's going to set up the baseline. That's going to calibrate the machine to give you the 0 observance baseline, right? So you use this sample as your baseline cuvette. Okay, and the rest of them will give you the observance. Okay, once you have these values observed from your 5 samples, what you're going to do is you're going to create a standard curve using Excel. So enter the amount of concentration of BSA on one column, and then A595, this is visible light of the observance on another column, and then create a line graph. Okay, on a graph, and then you figure out its best fit line and get its R-scale value and equation. Okay, so you need that equation to estimate the concentration of your unknown sample. Okay, so this table with these 6 diluents are needed, 6 diluents BSA are needed to construct a standard curve. Okay, from your standard curve you will get the equation, okay, linear equation, and that linear equation is needed to estimate the concentration of your unknown sample. Okay, after that's done, yeah, this slide talks about how you can create the standard curve using Excel, right? I just told you about it. 2 columns, concentration versus its observance. You can create that standard curve, and you get a best fit line and its R-scale value and equation. That's what you need. And if you have that, then you will be given with 2 BSA unknown sample. Okay, and you're going to estimate their concentration. How are you going to do that? First, you will have to make that dilution, maybe 1 to 100 dilution of each solution, and then from the diluent, you're going to take about 10 micro of each diluent and mix them with 790 microliter of water. So that's going to give you 800 microliter, right? And then add 200 microliter of breakfast dye solution to make 1000 microliter. Okay. You also need a blank cuvette, which will just have nano pure water 800 microliter and then this dye solution, because you need to calibrate the machine. Okay. And these two samples will be read at A595. So you will get the observance, right? Then what do you do with the observance? So you're going to get that observance. Observance will be your Y values, which is the dependent variable. Is that right? So you plug that observance value into the equation you're going to get from your standard curve. Then you can solve the equation for the unknown value. In that case, the unknown value will be the concentration, right? So the concentration can be mathematically calculated using the equation you got from the standard curve. That's how you can determine the concentration of the protein samples. Okay, so that part should be done. All right, so based on this, you will be writing the lab report 3. Lab report 3 is about protein concentration only. DNA, RNA, yeah, you can include that if you want to. The main part is the protein concentration with the standard curve, best fit line, trend line, and then R-square value and equation. And then based on that, your estimated value of BSA unknown A and B sample, their concentration estimation will be your main data. Okay, so we're going to talk about that during the lab face-to-face as well.